Okay, so your electron transport chain is it's basically a series of complexes that you find in the inner mitochondrial membrane. Right. The whole purpose of this chain is to shuttle electrons towards oxygen and create water, at the same time creating a proton gradient using your hydrogen atoms to then produce ATP as the final goal through ATP synthase. So I'll probably like refresh your memory a bit. Do you remember we were talking about glycolysis? Then from glycolysis we talked about pyruvate decarboxylation. Then from pyruvate decarboxylation we talked about the Krebs cycle. You remember that? And
Yes.
basically, all those things they make up something called oxidative phosphorylation, which is the process by which your cells generate ATP. Right.
Yes.
So if you look at how glycolysis and how the Krebs cycle were working, for glycolysis, for example, at some point you did have ATP being produced, but that ATP is not enough to sustain the body. So the whole purpose for glycolysis, we wanted to get pyruvate, then we decarboxylate the pyruvate and we channeled it towards the Krebs cycle. The Krebs cycle then produced what are known as high energy electron carriers. And the high energy electron carriers that we are talking about, it's NADH and FADH. So you have NADH, then you have FADH2. So these are the high energy um, electron carriers that you're getting from the Krebs cycle, right? So what then happens is, if you remember, glycolysis was a process that was happening um, is a process that was happening inside the cell, but you're talking about the cytoplasm. But here now we want to talk about a process that's happening on the inner mitochondrial membrane, where you have these four complexes. Complex one, this is called NADH dehydrogenase. I think it's the easiest word to use for complex one enzyme. It's called NADH dehydrogenase. Right. Others prefer to call it NADHQ oxidoreductase, but let's just call it NADH dehydrogenase. What this complex does is it accepts two electrons from NADH and you produce something called NAD plus, right? So this NADH, you remember it's coming from glycolysis, it's coming from
Yes.
pyruvate decarboxylation, it's coming from the citric acid cycle, right? So this kind of a reaction is an oxido reduction reaction in the sense that there are things that are gaining electrons, then there are things that are losing electrons. Remember in chem we used to say oil rig. Oxidation is loss of electron, reduction is gain in electron. Right. So here you can
Yes.
see what's labeled in blue there. That's your so that's basically your your electrons, right? So complex one accepts the electrons from NADH. And the whole purpose for this is this complex will transfer the electrons to something called coenzyme Q. Yeah. So it's called coenzyme Q. It can also be called ubiquinone. So coenzyme Q, or you can call it ubiquinone. Right. But if you remember from chemistry, where electrons are moving, you generate a current, right? So as you pass electrons through the electron transport chain, as they're moving down, right? Protons are going to be pumped, and these are your H plus ions. They're going to be pumped into something called the intermembrane space. So intermembrane space is simply the space that is between the two membranes of the mitochondria. So mitochondria has an outer membrane and an inner membrane. Right. So you're going to pump the protons um, into the intermembrane space. And those protons that you are concentrating in that space, they're going to create a very, very important gradient that we're going to use to produce ATP in the end. Right. So the first thing, we had complex 1, which we called NADH dehydrogenase. It accepted the electrons from NADH. Then it transfers those electrons to coenzyme Q, better known as ubiquinone. Then the second complex here, it's called complex 2. It can also be called succinate dehydrogenase. So remember when we started, we said we have two um, high energy molecules that we are bringing here, NADH and FADH2. So what succinate dehydrogenase does is it accepts the electrons that are coming from FADH2. And you produce something called FAD, or just FAD plus. So complex 2 will transfer the electrons from FADH2 and it will transfer them to ubiquinone, or your coenzyme Q. So basically what's happening is complex 1 and complex 2 are accepting electrons from NADH and FADH2 respectively. Then you transfer your electrons to coenzyme Q. Right. Then there's another complex that is in between that is called complex 3. So complex 3 can also be called cytochrome BC complex, but let's just call it complex 3. What complex 3 does is it transfers electrons from coenzyme Q, which got electrons from complex 1 and complex 2. It transfers those electrons to another enzyme known as cytochrome C, which you can see in this diagram being green, right? So cytochrome C will get the electrons. But always remember, as you're moving electrons down, you're producing what? You're producing a current. And what are you using? You're basically using what? H plus ions. So you're pumping more and more H plus ions into the intermembrane space. Then now electrons are now with cytochrome C. The last complex here is complex 4. So what complex 4 does, it's also known as cytochrome oxidase. What complex 4 does is it is the final electron acceptor. So it will take the electrons from cytochrome C and it will transfer them to oxygen. And as you do that, you produce water, which is the H2O. So that is the very reason why anything that happens in oxidative phosphorylation as a whole, you need oxygen to be there. If you're in a hypoxic state, ATP production becomes low. Why? Because the final electron acceptor in all this is going to be the oxygen. So complex 4 will transfer the electrons from cytochrome C to oxygen, then you make water. But then what has happened so far is the electrons were being moved from complex 1 to coenzyme Q, from complex 2 to coenzyme Q, then from coenzyme Q to complex 3, up all the way to complex 4, where you eventually made water. You were pumping protons into the what? into the intermembrane space. So why is that important? There is a very, very crucial enzyme called ATP synthase also on this what? On this inner mitochondrial membrane. And what this ATP synthase does is, since hydrogen proton ions are now more on one side, it is going to use that gradient to pump the H plus ions in the opposite directions. But as you're pumping H plus, remember we're calling it an ATP synthase. It's just like an ATP H plus pump. So as you're pumping your protons, you're going to have production of ATP. Right? So what basically happens is you're going to have ADP coming in contact with an inorganic phosphate, then you make ATP. Right. So what has basically happened is the four complexes have helped us create a proton gradient. Then now a very important pump, which is your ATP synthase, is the one now that is going to combine ADP and inorganic phosphates to make ATP at the expense of moving hydrogen ions or your H plus ions in the opposite direction. Right. So for this whole thing to work, the most important thing here is oxygen availability, because oxygen is the final electron transporter. Then also, if you're in a high energy state or high energy demand, you're running, you're doing activities that need more energy, that is going to propel this electron transport chain to work more and more, right? So for cellular respiration to suffice or to actually continue, this this is a very, very important step, right? So now let's talk about what has happened before. Remember, I said you're having basically the high energy molecules coming from glycolysis, coming from pyruvate decarboxylation, coming from the Krebs cycle, right? So a single NADH molecule that comes from the citric acid cycle will give you 3 ATP molecules, whereas a single FADH2 molecule that comes from the citric acid cycle will give you only 2 ATPs. The way I remembered it when I was in part 1 was FADH2 will give me two what? Will give me two ATPs. Then NADH will give me one more. And the one more is what? Is and the one more is basically 3. Right? So in a nutshell, the electron transport chain is there to generate a gradient that will be used by ATP synthase, and ATP synthase will then produce your ATP, right? So if you then want to look at these complexes one by one, you realize that, for example, in complex 1, there's a molecule called FMN, or better known as flavin mononucleotide. So that flavin mononucleotide that is in complex 1 is the one that accepts those electrons that are coming from NADH. Right. So far, so good.
Yes.
Okay, great. So it means you need to have glycolysis, pyruvate decarboxylation, citric acid cycle. You need to have them on your fingertips. Then you then understand what's happening in electron transport chain. So there's a question that I like asking. glycolysis as a process would give us about two ATP molecules net result, which is less. But it gave us so you also get like NADH molecules from glycolysis. But the NADH molecules that are coming from glycolysis, right? They don't produce the same number of ATPs as the NADH molecules that are coming from the citric acid cycle. Why? Because of location, right? So it's different. So do you by any chance remember where glycolysis occurs? It's in the cytoplasm, right?
Mhm.
Yes. So glycolysis occurs in the cytoplasm. Then now you want to push that NADH coming from the cytoplasm into the inner mitochondrial membrane. So it has to pass through the membrane, the gradient, so it has to use energy. So overally, the NADH that will be coming from glycolysis will produce fewer ATP molecules compared to this to the citric compared to the NADH molecules that are coming from your citric acid cycle, right? Why? Because your citric acid cycle occurs inside the mitochondrial matrix. So those NADH molecules do not have a distance to move because they are simply going onto the complexes that you're finding on the inner mitochondrial membrane. So it becomes a bit different, right? So in a summary, complex 1, NADH dehydrogenase, accepts electrons through FMN, known as flavin mononucleotide. Then it will later on transfer the electrons to coenzyme Q, also known as ubiquinone. Then complex 2, better known as succinate dehydrogenase, will accept electrons from FADH2 and make FAD. It will transfer the electrons also to coenzyme Q, better known as ubiquinone. Then from coenzyme Q, the electrons will be shunted by complex 3 to cytochrome C. Then complex 4, which is your cytochrome oxidase, would then transfer electrons from cytochrome C to oxygen, which is the final electron acceptor, right? And you produce water. And as you're moving your electrons through the electron transport chain, you're pumping protons into the mitochondria intermembrane space, so the mitochondrial space between the two membranes. Right. So the gradient which is then produced, right? The gradient that is then produced is the one that is then going to produce ATP. So there's a process which is called chemiosmosis. I think you remember that from chemistry, where your hydrogen ions are moving selectively uh, across a membrane. So you're just having hydrogen ions moving and creating that gradient, right? So you then produce three ATP molecules from a single NADH molecule. Then a single FADH2 molecule will give you what? Will give you two ATP molecules. And what basically regulates this electron transport chain is oxygen availability, which is the most important thing. Number two, energy demand. There are other things like the ratios of the NADH molecules, FADH two molecules versus their reduced components, but well, that's something else. But what's really important is oxygen availability and um and your energy demand, right? So ATP synthase then becomes the most important enzyme across this whole thing here, right? Why? Because it is the one that is going to convert the energy or the potential energy that you have in the protons into ATP. So ATP synthase becomes the most important enzyme in this complex, though it is not really a part of the protein complexes that you have in the chain. So you only have 4 protein complexes, then ATP synthase being the final thing that then produces your what? That then produces your ATP. So do you have questions on the electron transport chain? So biochemistry, I like people who just look at a picture like this and you describe it that way. Going to a book, it's a waste of time because you have everything in one picture and you have to make sense of it in one go. Right. So do you have questions
No.
before we move on to oxidative phosphorylation, which basically is still basically going to be summarizing this again? Suppose you'd find it's almost the same thing which we are going to be talking about. Just that by definition it will be um slightly different, but it's still the same thing. So any questions so far?
No.
No questions. Great. So we can now try to do oxidative phosphorylation.